welcome everybody to one of the most important grand rounds of the year. This is the Trainee Research Award presentations to celebrate and showcase some of the most important research and some of the best research that's being done in the department. And it spans the gamut. And so for that, I'd like to thank the committee. We also have to have a number of people doing different kinds of research to explain to each other some of what this means. Thank my co-chair, Eric Hung, and then the members of the committee, Andy Crystal, Marina Tulasham, Steve Hinshaw, Dan Mathalon, Dave Manoli, and I hope I didn't forget anybody. The one thing I will ask you to do is to please put your questions in the public chat. And just to remind everybody, it's a very difficult to choose from some great applications, but please keep them coming for next year. Think of it, we particularly want people that are in their final year, priority for people who are senior researchers in their final year, and for work that's being done at UCSF. So thank you very much, and I'm going to allow Alyssa Hamlet to introduce Alyssa Hamlet.
Thank you so much, Susan, for your work. It's always such a pleasure to meet the trainees in our department. So it is my pleasure to introduce Alyssa Hamlet this morning. She I think our department discovered her about 15 years ago when she had just graduated from UC Berkeley in cognitive science. And she was very clear and focused on her goals. And she wanted to really know if she liked research, mental health research. So she entered the labs of Owen Wolkowitz and myself and worked for many years, moonlighting for us, doing research, and decided this was her path. So she got a PhD in clinical psychology at Temple University with Lauren Alloy. She got an NSF award there for her research. She studied depression, cognitive functioning, memory, and there she started her long-term interest in pubertal timing and major depression and the development of high risk for depression in girls. And also she looked at puberty in boys and especially examined race. She then did a postdoc at University of Illinois, and she was absolutely prolific there and became one of the most published authors on this relationship between early life depression and early pubertal timing. And then she came to us, and we got very lucky to include her in our T32 program. She's in her second year, and not surprising to me, she's entered with tremendous curiosity to really understand not just sexual reproductive development and history, but all of aging biology. She's kind of taken on and said, I'm interested to understand what's happening at the cellular level too. She's already published several papers on the epigenetic clock. We'll hear more about that today, and has been working closely with Steve Horvath, who's been pioneering that. So there's been it's long been the question to understand the intertwining relationships between the biology of aging and mental health and depression. And Alyssa has been looking at that across the lifespan and has just an exciting program of research that I will let her tell you about. Thank you, Alyssa.
Thanks, Alyssa. I'm really honored to be here today, and thank you so much, and to share my work with you. So I know we're a little tight on time, so I will go ahead and share my screen. So I'm going to talk to you today about my work on early life adversity and pubertal timing, and how they can both confer risk for epigenetic age acceleration. And I have a brief agenda. It's pretty much, we're going to hit them 1, 2, 3. First, I'm going to talk about why early life adversity is an important thing to be studying if you're concerned with mental and physical health outcomes across the lifespan, and how early life adversity can then confer risk for early pubertal timing or maturing earlier than your peers. And lastly, I'm going to show how early life adversity might actually start getting under the skin at an early age and start to affect biological aging, which could be a mechanism by which it affects health in later life, and how pubertal timing might actually be kind of maybe a special case of an early form of biological aging. So when I say early life adversity or ELA, I'm going to be saying that a bit today. I'm mostly talking about that within the ACE framework, with its adverse childhood experiences. And those are usually thought of in terms of three broad categories of abuse or neglect or household dysfunction, which is something that might not directly affect a child, like a relative being incarcerated, but that certainly might have a negative indirect effect. And ACEs have been really salient recently because there has been a lot of good, robust evidence that experiencing these type of events before the age of 18 can dramatically increase the risk of poor mental and physical health later in life. I think 9 out of the 10 leading causes of death are, you have an increased risk if you've had an ACE, or certainly numerous ACEs. And the Surgeon General of California has really been a leader in having primary care physicians screen for these events to identify those who might be at greater risk. And indeed, having ACEs can even increase your risk for premature mortality. So it's interesting that these very early events can have very severe lifelong consequences. This is just one area. I have, from the beginning of my career, been very interested in depression as an outcome. And this is just showing you that being diagnosed with depression, these are California adults, that the risk increases pretty continuously with the number of ACE events you have experienced. But I'm also really interested in early adversity because, I mean, in and of itself, it's worthy of study, given that it has an effect on all these outcomes. But it also has a relationship that has been found with puberty and pubertal timing. And a lot of this comes from life history theory, specifically the psychosocial acceleration hypothesis. And basically, it holds that in a risky or adverse early environment, the organism the body gets a signal that delaying reproduction and waiting to have offspring when you're more ready might be a lot riskier than going ahead and reproducing as early as possible before you get put out of commission by death or disability, and therefore aren't able to pass on your genes. And there has been some empirical evidence. There still needs to be more, that especially in girls, maturing or having this early life environment that has uncertainty and risk might lead to an earlier pubertal maturation. So you're kind of accelerating your reproductive capacity to be able to do it as early as possible. And we don't know exactly how this happens, but it probably has to do with some kind of recalibration of the HPA axis and its relationship to the HPG axis. I'm also going to say pubertal timing a lot, and it's simply the level of secondary sex characteristics that change during puberty compared to an individual of the same age and the same sex assigned at birth, usually. So if I'm talking about an 11-year-old girl, I'm comparing her development to another 11-year-old girl. And these are usually, a lot of times they're studied categorically, and kids are kind of bifurcated or trifurcated into being earlier than your peers, being later than your peers, and being on time, maturing at the same average rate as most of your peers. I also like to study it continuously when possible. And pubertal timing in and of itself, or maturing earlier than your peers, has also, like ELA, been associated with poor mental health and even poor physical health. And especially for girls, the evidence has been somewhat robust, especially for depression. And this is a paper I worked on not too long ago, where I actually found that early pubertal timing conferred risk for the first onset of depression during adolescence, but it also conferred risk for the recurrence of a second episode during adolescence within a period of 3 years. And I actually found that for girls and boys. And so though a lot of work shows early timing to be riskier for girls, I'm also interested in boys because boys are actually understudied in pubertal research. And also it's a little bit harder sometimes because you don't have the kind of salient reproductive milestones like the onset of menstruation. This is another thing I worked on where I did, part of that was a lit review. And so even though my primary interest has been depression, I found that early pubertal timing, maturing earlier than peers, actually might be a transdiagnostic risk factor. And so I looked at the lit on pubertal timing, early timing, and the relationship to different forms of psychopathology at both the disorder and the dimensional level. If there's a check, that means there was a positive, relationship found between early timing and that form of psychopathology. Unfortunately, gender normatively, I have pink for girls and kind of a gray blue for boys. An X is null evidence. And although you can see there's probably some publication bias and there's some areas that are understudied, the overall pattern is that early pubertal timing looks like a risk factor for both internalizing and externalizing problems. Most of these are in adolescence or childhood, not in adulthood yet. We don't have as much data on whether it's a risk factor for later psychopathology. And then it does look like it could function similarly for both boys and girls. Okay, I'm going to contradict myself a little bit, but most of the evidence shows that girls do have a more severe outcomes when it comes to early pubertal timing. Like it can cause issues for boys, certainly, but if you're looking at clinical levels of psychopathology, you're looking at the onset of depressive episodes, it most clearly looks like girls are at greatest risk when they mature earlier than their peers. And that might not be too surprising because on average, girls mature about a year earlier than boys on average. So if we're saying early is bad, then if you're the earliest of the early, certainly that might be especially risky. And this is just a very recent paper I found. And it's age at menarche, so it's age at first menstruation. If you're early, the consequences are on the left. And if you're later, the consequences, like later than average, the consequences are on the right. And I'm not going to go into all these details, but I'm just showing that early maturation appears to confer a risk for quite a few physical conditions as well as psychological issues in girls. So I'm just kind of showing how it seems to have this pubertal timing seems to have a pretty broad impact on health, similar to ELA. And then I talked right before that just how early life adversity can actually function as a risk factor for early pubertal timing, or so we think. But I'm kind of interested in how this would happen. Like, what would be the mechanism by which early life adversity would actually make a child more likely to develop these problems much later in life. And I became interested when working with Alyssa here in biological aging. And so how this early life adversity might actually get under the skin and to confer risk for these conditions later in life. And I'll talk about the exact relationship to pubertal timing, but this is kind of the study design that has informed my work here that I'm going to present the meat of this study. So when I say biological aging, I'm talking about what your cells tell us compared to your chronological age. And it's kind of a general term, biological aging. but there's several ways that you can measure it. And the way that I have chosen to do so has been primarily using the epigenetic clock, which is based on looking at the methylation of certain sites in our genome that have been kind of in a data-driven approach correlated with chronological age, and then looking at people's patterns and how they line up with those patterns we know that correlate with age. So what we're basically looking at here is called age acceleration, and that's your biological age compared to your chronological age. So if you're the guy above the curve here and you have a positive age acceleration, that means your chronological age is less than your biological age. So you might be 40, but your cells are saying 50. And that is going to be associated with worse health outcomes, whereas the guy below the curve here has a negative age acceleration. He is actually biologically younger than his chronological age. So he might be 40, but his cells are saying, you're 35. And so there's been a lot of exciting research. Steve Horvath, Alyssa mentioned, that has been looking at what has been associated with age acceleration. And there's been such an amazing amount of mental and physical health outcomes that are consequences of having being biologically older than your chronological age. And indeed, major depression has been one of them. But it's really an exciting area of research. And so I began thinking about, okay, so is this related to pubertal timing? Is pubertal timing maybe a mechanism itself by which early life adversity, early life trauma confers risk for biological aging, thereby increasing risk for health conditions? And so it was really important to look at that in a group of relatively young, relatively healthy sample. And so this was in 183 women. They were all around the same age. They were all premenopause, and early 40s was the mean. And they were a pretty homogeneous sample in that they were primarily white, but most importantly, they were all non-smokers. They had a BMI that was under 40, and there were no major diseases. And that's an important thing to account for when you're looking at epigenetic aging. And so I looked at the effect. What I was kind of positing here was that there was an indirect effect of early life trauma on epigenetic aging, and that it operated through pubertal timing. And I further looked at early life trauma in the domains of both abuse and neglect, because there's been some evidence that abuse is the most important for epigenetic aging. And for pubertal timing, I actually looked at age at first menstruation. That's one way that is very kind of the quick and dirty way of looking at pubertal timing in women. And so the idea was that early life trauma having more trauma would confer risk for earlier age at first menstruation, which would in turn accelerate epigenetic aging. And I looked at epigenetic with aging with DNA Grimm age, which is kind of the newer generation of epigenetic clocks. And I especially like this clock because not only does it correlate well with chronological age, it correlates with outcomes I'm really interested in that it predicts very well age at death, time to death. It predicts time to heart disease and time to cancer. And these outcomes are really important for, and they can be consequences of ELA as well as early pubertal timing. So what did I find? I did not find the indirect effect, which was a little surprising, at least at first. I found two pretty robust main effects in that early trauma, specifically early abuse. It was not found with neglect. Having more incidences of trauma before age 18, and this is epigenetic aging in adulthood, remember, conferred, was associated with being biologically older than your chronological age. Similarly, maturing early and having an early age at your first period was also strongly associated with being biologically older than your chronological age. So this is a really interesting way to say that both early life and puberty are really these kind of sensitive periods, at least for accelerated biological aging. And these could both be really interesting periods to intervene for this health trajectory of girls in a multitude of areas. Now, I did not find the relationship I thought between trauma and pubertal timing. And so that kind of made me think a little bit more about how I was conceptualizing these two constructs of pubertal timing and accelerated biological aging. I just want to say briefly as an aside that one of the kind of cornerstones of my research project has been to look at pubertal timing in a diverse group of girls, because the history of the research has usually been in white middle-class girls. And so I have made my grad school career was all about looking at pubertal timing in Black girls as well as white girls. But here at UCSF, I've had the opportunity to look at early life adversity and how that might affect puberty in both Black and white girls, and that I've had access to this amazing sample of equal numbers of Black and white girls that were assessed from age 9, 10 in the late 80s for 10 years with kind of the gold standard of puberty assessment, which is the medical exam by a professional. And we also have these girls are being followed up now with by me and Alyssa and some of our collaborators here and at UC Berkeley. And so we have measures of early life adversity as well. And so I just basically wanted to see if race moderated this relationship between early life adversity and pubertal timing. I also looked at pubertal tempo as an outcome, which is another way of looking at individual variations in puberty. It's simply the rate through puberty. Are you fast? Are you slow? So I had about 375 kids, half Black, half white, and we looked at the number of ELA, the number of events before age 12, and then I had growth curve models looking at their development from ages 9 to 16. And I looked at pubertal timing at age 10. So, and it's really important too, because BMI and puberty and has a really strong association. I had BMI as a time varying covariate. So I was taking into account their BMI at each time point. So what did I find? This is under review right now. So it hasn't been published, but basically I found that ELA affected pubertal timing, in that the more ELA you had, the earlier you were at age 10, and you also had a slower tempo from 9 to 16. But I only found this relationship for the Black girls. And I actually found no relationship between pubertal timing and tempo with either income or education or other measures of SES. So I'm still kind of trying to understand exactly why this relationship would only be significant for the Black girls. It wasn't the right, same direction with the white girls, but it seems like this might have something to do with the weathering phenomenon, which kind of holds that, especially in Black women, just being in an environment of disadvantage, or an environment that is difficult, might confer risk for higher allostatic load. So kind of like a biological aging. And so maybe this is happening at a really young age. Maybe being a Black girl is an adverse or risky environment in the US. And, that is somewhat aligned with the psychosocial acceleration hypothesis. And so maybe if you combine that with ELA, you're going to see a more dramatic relationship with puberty. But this is by no means the end of the story. I'm just going to close by saying, so this is all of this work has gotten me to kind of think a lot about, is early pubertal timing just a specialized case of accelerated biological aging? I'm not the first one to think of this, and I've had some good talks with Jay Belsky, how these might actually be very similar processes, just occurring earlier in time than we usually think of when we think of biological aging. And right now with Alyssa, and I'm reading a grant for it, I am working on looking at the role of race, but also looking at this question very comprehensively on how does early life adversity confer risk for depression, indeed, but also physical health comorbidities like heart disease and metabolic syndrome, and how those might operate through biological aging. Not just looked at by epigenetic aging, but actually looking at it as a terms of like a phenotype that would be made up of epigenetic aging, but also indicators like telomere length and increased inflammation at a younger age, and of course, early pubertal timing. So I'm kind of going to be looking at these as a aggregated whole. And not only do I have the opportunity to look at these in the NGHS women who are now in midlife, but I'm also going to have the opportunity to look at this relationship between ELA and health through this phenotype of biological aging in their children. And what's really cool is we have measures of ELA in these children that include stress of pregnancy stress of the mothers when they were pregnant with these children. And I think that's going to be a really cool way to see if this kind of early life adversity starts programming these children early in life to kind of have this accelerated aging, because in the short term, it's actually adaptive, right? You have the chance to reproduce. It's just that in our current society, it also, there might be a trade-off, especially if we're living longer, and it might have a trade-off in terms of longevity or having a shortened health span and having a more difficult disability and disease as we age. So, there seems to be a trade-off here. Okay, well, just all I have is my thank you. I just wanted to thank, of course, my mentor, Alyssa, without which none of this would be possible, and who has really believed in me from an early age. And I would also like to thank the members of the UCSF Center for Health and Community, which is a fantastic group I am very lucky to be a part of. And of course, funders, NIH in particular, who have really helped us with this line of work that's kind of still in its infancy.
Lisa, thank you so much. We have so many good questions for you, and we have a little bit less than 5 minutes for questions. So.
Oh, shoot, I'm sorry.
No, you were perfect on timing. So let me just get started with a really juicy question for you, which I know you've thought a lot about, which is how we might look in a more refined way at early trauma. So ACEs, the crudeness of ACEs, it's such a predictive measure, but it's also such a lump of different types of trauma. So do you think that different types of psychological trauma or adversity, SES, deprivation, et cetera, do they matter? Have you looked at different composites of types of exposures?
I mean, the most that I have done with it is I've kind of looked at the really excellent work by Katie McLaughlin and her group, in that she's really found abuse to be associated with early pubertal timing and accelerated biological aging with different indicators. She has not found that relationship for neglect in severely neglected or deprived populations, like we're talking the Romanian orphan studies. She's actually found neglect could be associated with later puberty. But again, most of the puberty work is done in first world environments. And so it's a very different, sometimes neglect has a very different way of being assessed than in different kinds of developing countries, which would be an interesting place to take this research and be internationally. But I also think that we might have to even look at the event level, because when just looking at the count of events, which actually is a decent way of doing it, the Black girls and the white girls in our study, although they reported events of ELA, they were very different events. And in severity wise, qualitatively, the kind of events the Black girls reported were, you could call them a lot more serious, and in some ways might have a more intense effect. So I do think that's a really question that needs to be worked on.
Thank you. Beautiful answer. That question was from Candace Black, who's joined us from University of Arizona, who is a trauma and health researcher. Next question from Karen Jones Mason, which is, could early trauma have intergenerational effects? Could you actually see it affecting puberty in the next generation?
Definitely. And that's what I think is so cool about this kind of study, because we have the women and we have stuff from when they were children, trauma from when they were children, but we also have a lot of measures about their environment that might be transmitted to their children, who are also now just entering puberty. So I think as long as I would like to continue to follow the women through aging, but also the children. And I do think this could be one way in which intergenerational trauma makes its mark. So, because it's been a little harder to study this if you don't have something quantifiable that you could measure, but biological aging might be a way. It's almost like a pre-programming this kind of phenotype that could be passed on in an environment of risk. And it'll be really interesting to tease out the nuances of that in our future work.
Yes. Juicy, another great question that's just
Talk all day about it.
have to be taken offline. Marty Horowitz's question about narrative memory. You know, some of our measures are retrospective, and so it might be based on, you know, on memory narratives, selective bias memory narratives versus actual prospective measures. Let's end, and then Lauren Frank has a great question about effect sizes. Let's end on a question that I think we're all wondering, which is from Tony Yang: treatment interventions. What are your thoughts? Is puberty at all malleable? What would we, you know, what are the targets that you're thinking of for interventions?
So interesting. I mean, I don't necessarily think that like delaying puberty is the answer, because I don't think early puberty has to be a bad thing. I think that a lot of my earlier work looks at the context in which early puberty takes place. And I think it's really how we treat these kids. I think in the right kind of environment, early puberty could even be a strength. I mean, there's kind of research that doesn't seem to be hold true, but in the 60s or 70s, early puberty seemed to be a strength for boys. And it doesn't look like it is that way any longer, but there might be environments in which we could change our environment and change the way we talk about puberty, change the way girls approach puberty, change the way parents talk about puberty in our society, and that might have an effect. I mean, we can certainly intervene. I mean, I think preventing ELA is the best thing we can do, as Nadine Burke-Harris has kind of set out to do. But I think that I really like that there's maybe a second opportunity. If, you know, we can identify these kids as being at risk, we then have this kind of window, a very plastic window, that could be a great time to intervene. And there's been some early interventions that might actually affect biological aging. And I think that's also something we might want to look at as a potential target. And also, I don't have time to answer that narrative memory question, but it's really, really interesting, especially because I have a big interest in autobiographical memory and memory bias. And at some point, I would love to combine that with this work. And so
Marty Horowitz, one of the treasures of our department.
Oh, I mean, I have to, thank you for that question.
In our department, we have Danielle Rubinoff, who's working on, you know, early adversity, Nicole Bush, Alicia Lieberman. So it's just so exciting to have such a great community here. Thank you so much, Alisa. Thanks everyone for your wonderful questions.
Thank you.